Welcome back to the deep dive. We are doing something a little different today.
Yeah, we're going microscopic.
We are. Usually we take a broad societal topic and, drill down, but today we are looking at a specific biological machine that breaks in a very, very specific way.
Or rather, 3 specific ways.
3 specific ways. We're tackling colorectal adenocarcinoma. Now, before you tune out thinking this is just some medical board review.
Which it absolutely is not.
No. I want to frame this differently. This is really a story about how complex systems fail.
It is. It's the ultimate lesson in systems engineering, or maybe systems failure. It's about how a perfectly good car ends up in a ditch.
Exactly. And looking at this stack of research you've pulled, you have papers from Vogelstein, the TCGA data, the NCCN guidelines. It really strikes me that colon cancer is a bit of a misnomer.
It is a huge misnomer.
We talk about it like it's one single disease. He has colon cancer. But based on these molecular profiles, it seems like we are talking about 3 completely different diseases that just happen to occur in the same organ.
That is the single most important insight to start with. It's fundamental. If you're a medical student, you learn colon cancer as this singular entity in your anatomy class.
Right, one chapter in the textbook.
But if you're an oncologist or a pathologist, you know that a tumor driven by the APC gene looks, behaves, and responds to drugs in a totally different way than a tumor driven by, say, microsatellite instability.
They might as well be different species.
They really might. It's like comparing a car, a boat, and a plane. They're all vehicles, but you wouldn't use the same mechanic for all 3.
And yet there's this cognitive bottleneck that the literature keeps mentioning. We see it all the time.
Oh, constantly.
People memorize the alphabet soup, APC, KRAS, TP53, SMAD4, but they can't string it into a coherent sentence. They know the parts list, but they have no idea how the engine works.
That's the classic reductionist trap in medicine. We teach students to memorize that APC is a tumor suppressor on chromosome 5. Great!
I can pass a test with that.
You can pass a test, but why does losing it cause a polyp? Why does it happen first? Why not last? Unless you understand the mechanistic storytelling, the why behind it, you're just memorizing phone numbers. It's useless in practice.
So that's our mandate for this deep dive. We're going to ignore the rote memorization, and we're going to build a functional model from the ground up.
From first principles.
And we found a framework in the prep notes that I really want to stress test with you today. It simplifies this whole process into 3 distinct mechanisms, using a car crash analogy.
The 3 cars model. Yes, it's a very useful heuristic. It's not perfect, of course, no analogy ever is, but it's fantastic for distinguishing the 3 major pathways.
Which are?
The APC pathway, the MSI pathway, and the CME pathway.
Right, so the framework is you can crash the car by cutting the brakes.
That's your APC pathway.
By breaking the spell checker in the car's navigation computer.
Your MSI pathway.
Or by someone hacking the engine's control software to shut down the safety systems.
The CIMP pathway. I might tweak that last one a bit. It's less about shutting down the engine and more about silencing the safety protocols, the airbags, the anti-lock brakes.
Okay, that makes sense.
But broadly, yes. It's brake failure, it's repair system failure, and it's epigenetic silencing.
So before we actually start crashing these cars, we need to understand the parts. We need a toolkit.
The cast of characters.
Exactly. And I don't just want the textbook definition. I want the mechanism. What is this molecule actually doing? Let's start with the big one, APC.
The gatekeeper.
We know it stands for Adenomatous Polyposis Coli. We know it's on chromosome 5q21. But mechanically, in a healthy cell at the bottom of a colonic crypt, what is this protein doing all day?
Okay, so to understand APC, you have to understand something called the Wnt signaling pathway.
Wnt signaling?
Yes. This is an ancient pathway used in embryology to tell cells, grow, divide, make an arm here, make a leg there. It's a fundamental growth signal.
And it's still active in adults?
In a very controlled way. In the adult colon, the Wnt pathway is what keeps the stem cells at the very bottom of the crypts dividing, so they can replace the intestinal lining, which sheds every few days.
So Wnt is the go signal.
It's the ultimate go signal. And the main messenger, the effector of that signal, is a protein called beta-catenin.
Beta-catenin. The builder?
The builder, the accelerator, whatever you want to call it. When beta-catenin levels get high in the cytoplasm, it does something very important. It translocates. It moves into the nucleus.
Okay.
And in there, it finds partners, transcription factors, specifically the TCF-LEF family. They link up, they bind to DNA, and they flip the switch on a whole suite of growth genes.
Like which ones?
The big ones are MYC and cyclin D1. These are master regulators that tell the cell, enter the cell cycle now, divide.
So high beta-catenin in the nucleus means the cell divides. Simple as that.
Simple as that. Now, obviously, you don't want your cells dividing constantly and forever. That's cancer.
Right, so there must be a way to turn it off.
There is. Nature evolved what we call a destruction complex. It's a beautiful little machine made of several proteins, axin, GSK3-beta, and our friend APC.
And APC is the key part of this complex.
It's the scaffold. It's the frame that holds this whole execution squad together. This complex sits in the cytoplasm, and its entire job is to grab any beta-catenin that's floating around, phosphorylate it, tag it, and mark it for ubiquitination.
And ubiquitination, that's the cellular kiss of death.
It's the cellular death sentence. It gets sent to the proteasome and just shredded into amino acids. So as long as APC is working, beta-catenin levels are kept incredibly low. The cell doesn't divide unless it's supposed to.
Until a Wnt signal comes in from the outside.
Precisely. When Wnt binds to its receptor on the cell surface, the first thing it does is trigger a cascade that dismantles the destruction complex.
So it breaks the execution squad.
It does. Beta-catenin is no longer being destroyed. It survives, it accumulates, it rushes to the nucleus, and you get controlled growth.
Okay, so let's complete the circle. If I lose the APC gene through a mutation,
The scaffold collapses. The destruction complex cannot form. Beta-catenin is never destroyed. It just piles up and up and up, floods the nucleus, and screams divide at the DNA 24/7.
even if there's no want signal from the outside world.
Exactly. The cell thinks it's constantly being told to grow, but it's really just a broken brake inside. The brake pedal is gone.
And that is why APC loss is the initiation step. It creates the very first lesion, the benign polyp.
You've got it. The cell is growing too much, but it hasn't learned how to invade or metastasize yet. It's just step one.
Wow. Okay, that's a much richer picture than just garbage disposal. Let's move to the next piece of the puzzle, KRAS.
The stuck gas pedal.
This is a term that gets thrown around at every single oncology conference. Is the patient KRAS wild type or mutant? Why does this one gene matter so much?
KRAS is what we call a GT-PACE. It's a molecular switch. It lives on the inner surface of the cell membrane. And when a growth factor like epidermal growth factor, EGF, hits its receptor on the outside of the cell, KRAS gets activated. It does this by binding to a molecule called GTP.
So GTP means on.
GTP means on, and once KRAS is on, it passes that growth signal down a bucket brigade of other proteins. You've heard the names BRAF, MEK, ERK, all the way to the nucleus to, again, promote cell division.
So it's a relay runner in the growth race.
A perfect analogy. But here's the crucial catch. KRAS has a built-in timer. It is supposed to hydrolyze that GTP back into GDP.
And GDP means all off.
GDP means off. It's a self-emitting switch. It's designed to fire once, pass the message, and then reset itself immediately.
So it's supposed to be a quick pulse of signal.
Exactly. But in cancer, you get a single point mutation, a tiny typo. Usually at codon 12 or codon 13, you'll see names like G12D, G12V.
Okay.
This one tiny change in the protein's shape is just enough to break its timer. It jams the off switch. KRAS can bind GTP and turn on, but it loses its ability to turn itself off.
So the relay runner just keeps running laps around the track without ever waiting for the baton.
That's it. It drives that downstream MA penase pathway constitutively, meaning constantly. And that's why we called it undruggable for so many years.
Why was that?
It's basically a smooth sphere. There was nowhere for a drug molecule to grab onto to stop it. That is, thankfully, finally changing now with new G12C inhibitors, but for decades it was a huge challenge.
That explains the gas pedal analogy perfectly. Okay, next up, let's talk about the guardian, TP53.
P53. Probably the most studied gene in all of history.
The police officer of the cell.
That's a great memory hook. It's a transcription factor, but it's a very special one. It acts as a stress sensor.
A stress sensor.
Yes. In a normal, happy cell, P53 levels are kept very low because another protein, MDM2, keeps destroying it. But if the cell detects stress DNA damage from radiation, from chemicals, from oxidation,
The alarm bells go off.
the alarm bells go off and P53 stabilizes. It stops being destroyed. It forms a tetramer. 4 of them link up, and this complex binds to DNA.
And what does it tell the DNA to do once it's bound?
It basically makes a choice. It has 2 main programs. Program 1 is cell cycle arrest.
Hitting the pause button.
Exactly. It turns on a gene called P21, which is like a clamp on the cell cycle machinery. It says, nobody divides, stop everything while we fix this damage.
And what if the damage is too bad to fix?
Then it initiates program 2, apoptosis. It activates genes like BAX and PUMA, which are pro-suicide genes.
Through suicide, wow.
They essentially go to the mitochondria, punch holes in them, and trigger the cell to self-destruct in a clean, controlled way.
So P53 is the ultimate decision maker between let's repair this mess and let's take one for the team.
It is the judge, jury, and executioner. So now just imagine what happens when you lose both copies of your TP53 gene.
The police officer's gone.
The police officer is gone. You have DNA damage accumulating. You have oncogenes like KRAS firing like crazy. But there is no arrest. There is no suicide. The cell just keeps dividing, accumulating more and more messy, broken chromosomes.
And that leads to the genomic chaos we see in advanced cancers.
It's the final gate to true malignancy, in many ways.
Okay, we have one more major player to define before we can start crashing cars. The mismatch repair system, or MMR.
The spell checker.
The spell checker.
Yeah.
Now, we need to be very specific about what it's checking for.
Yes, this is important. DNA polymerase is an absolutely amazing enzyme. It copies billions of base pairs with incredible accuracy. But it has an Achilles heel. It struggles with microsatellites.
So explain microsatellites again. What are they chemically and why are they so hard to copy?
A microsatellite is just a short, repetitive DNA sequence. Imagine a sequence that's just cytosine adenine repeated 50 times in a row. Kaka?
Okay, a simple stuttering sequence.
Exactly. Now, when the polymerase enzyme is zipping along, copying this, the two strands of DNA can temporarily breathe. They separate and reanneal.
Right.
Because the sequence is so repetitive and monotonous, the strands can slip. They can reanneal in the wrong place, creating a little loop of extra DNA that bulges out.
It's like losing your place when you're counting identical tiles on a floor. You skip one or you count one twice.
That is the perfect analogy. This is called polymerase slippage, and it happens constantly in these regions. That's where the MMR system comes in.
Spell checker.
The spell checker. It's a group of proteins, MLH1, MSH2, MSH6, PMS2, that basically ride along behind the polymerase. It recognizes that little loop, that bulge, it clamps onto it, chops out the error, and tells the polymerase to try again.
So it's constantly cleaning up these little typos.
Constantly. So if your MMR system fails...
The loops stay, the typos accumulate.
Exactly. You get insertions or deletions of these little repeat units all over the genome. And that phenomenon is called microsatellite instability, or MSI.
Okay. We have our toolkit. We have the destruction complex, APC, the stuck switch, KRAS, right, the guardian, P53, and the spell checker, MMR. Now let's put them into motion. Let's build our first crash scene. Pathway 1, the classic pathway.
This is the pathway described by Bert Vogelstein in his huge, seminal 1990 paper. It's often called the Fearon-Vogelstein model, or the adenoma-carcinoma sequence.
And this accounts for the vast majority of cases, right?
It does. About 80% of sporadic colon cancers follow this route.
This is our broken break scenario.
Yes. And the molecular hallmark of this pathway is something called chromosomal instability, or CIN.
So what does that mean, chromosomal instability?
It means if you were to look at the karyotype, the picture of all the chromosomes from one of these tumor cells, it's a complete horror show.
It's not the Need 46 we learned about in school.
Not even close. They might have 60 chromosomes or 30. They'll have chunks of chromosome 18 fused onto chromosome 8. There are massive deletions and amplifications. It is gross structural failure. The car's chassis is bent and broken.
And the sequence of these failures matters. The mnemonic from the source material was AKSP, a king sits proudly.
It's a very useful way to remember the progression. It's a sequence of genetic hits.
So let's walk through it. First, A.
A is for loss of APC. This is the initiating event. It happens in a single stem cell at the base of a crypt. You get that second hit, lose your one good copy of APC. The destruction complex fails, beta-catenin rises, and the cell starts to divide more than its neighbors. This creates what we call a dysplastic aberrant crypt focus.
The very first microscopic lesion.
The very first thing you could see under a microscope that looks wrong. It's a small, benign adenoma.
Then we get to K.
K is for KRAS activation. The polyp is growing, but to become more aggressive, it needs to drive that growth even faster. A cell within the polyp acquires a KRAS mutation.
So the gas pedal gets stuck down.
Exactly. Now the polyp grows from a small adenoma into a large one. It gets more disorganized. Pathologists call this a tubular adenoma, or if it gets more complex, a villus adenoma.
Okay, so we've gone from a small bump to a larger, more worrisome polyp. Next is S.
S stands for SMAD4, or more broadly, the loss of the TGF-beta signaling pathway.
The red light.
The red light. Normally, TGF-beta is a signal that comes from the surrounding normal tissue, the stroma, and it tells epithelial cells to stop growing. It's an inhibitory signal,
A break from the outside.
a break from the outside. The tumor cells, to become truly independent, need to learn to ignore that signal. They do that by mutating a key component of the pathway, often SMAD4. Now they don't care what their neighbors are saying.
They've cut themselves off from the community's rule.
That's a great way to put it. And finally, we get to P.
P for P53.
This is usually the final critical step. This is the transition from a benign, albeit large, adenoma into an invasive carcinoma.
The police officer is gone.
Once P53 is gone, the genome just becomes catastrophically unstable. You get that loss of heterozygosity we talked about. The cell can now amplify oncogenes and delete tumor suppressors wholesale. It can survive massive DNA damage. It's the point of no return.
And this whole AKSP sequence, this is the story of the classic left-sided colon cancer, typically.
Typically, yes. These tumors tend to form in the distal colon, the sigmoid colon, and the rectum,
And because of the anatomy of the left colon,
which is much narrower than the right side, and the stool is solid by the time it gets there.
the tumor grows in a specific way.
It grows in a ring shape, it's annular, it infiltrates the wall and tightens around it like a belt.
The classic napkin ring sign you see on an imaging scan.
Exactly. And that constriction of the lumen, that tightening, is what causes the symptoms.
Which would be what?
Obstruction, cramping, colicky pain, pencil-thin stools, a change in bowel habits. The biology dictates the anatomy, which dictates the clinical presentation. It's a beautiful, logical progression.
It's so satisfying when it all lines up like that. But now, let's shift gears completely. Let's look at the second way to crash the car, the MSI pathway.
A totally different beast.
This feels fundamentally different because we aren't talking about smashing whole chromosomes. We're talking about tiny, subtle errors.
That's the key distinction. In the APC pathway, you are losing or gaining whole chunks of DNA. In the MSI pathway, the genome is actually diploid or near diploid. It has the right number of chromosomes. But the sequence of the DNA is riddled with thousands upon thousands of typos.
The typo explosion.
It creates what we call the mutator phenotype. The background mutation rate in these MSI tumors is 10 to 100 times higher than in the chromosomally unstable tumors.
But here's the question that always bugged me. Why does a typo in a repeating sequence, a kakaka, actually cause cancer? I get that it's messy, but why is it malignant?
That is the critical question, and the answer is that evolution has played a really nasty trick on us.
How so?
It turns out that several key tumor suppressor genes just happen to contain these microsatellites, these repetitive sequences, right in the middle of their coding regions.
So they have these built-in slippery sequences right in their own blueprints.
They do! It's a terrible design flaw. The most famous example is the gene for TGF-beta.
That's a receptor for that TGF-beta red light signal we just talked about.
The very same one. That gene has a sequence of 10 adenines in a row, and A10 repeat right in its coding region.
A. That sounds like a nightmare for a DNA polymerase.
It's a nightmare. It slips on that sequence constantly. In a normal cell with a working MMR system, the spell checker comes along and fixes it every single time.
But in an MSI cell,
the polymerase slips, you get an A9 or an A11 sequence instead of A10, and that tiny change creates a frameshift mutation.
what does that mean, a frame shift?
The DNA code is read in groups of 3, like 3-letter words. If you add or delete one letter, the entire rest of the sentence becomes gibberish.
So all the downstream code is misread.
Completely misread. You usually generate a premature stop codon, the protein is truncated, and it's just garbage. It gets degraded.
So you lose the red light receptor, not because you deleted the whole chromosome it was on, but because of a single typo.
A single unfixed typo. And it's not just TGF-beta. Another major target is BAX, a pro-apoptotic gene.
A P53 target.
Right. It has a G8 repeat, polymerase slips, you get a frame shift, BAX is gone. Now the cell can't commit suicide properly, so you get increased growth from losing the red light and decreased cell death from losing the suicide signal. A double whammy.
Now, we have to make a distinction here that the outline alluded to. MSI is the result. But what is the cause? Because I know this is where Lynch syndrome fits in.
This is a crucial clinical distinction. You can develop MSI in a tumor for one of two reasons.
Okay.
Reason 1, you were born with a broken spell checker. You inherited a mutated non-functional copy of an MMR gene, like MLH1 or MSH2, from one of your parents.
This is Lynch syndrome.
This is Lynch syndrome, also called hereditary non-polyposis colorectal cancer, or HNPCC. Every single cell in your body is already halfway to becoming cancerous because it's missing one of its spell checkers.
That's the germline defect. You're born with the first hit.
Right. But reason 2 is actually much more common. It accounts for about 12% of all colon cancers, whereas Lynch is only about 3%.
And that is?
You were born with perfectly normal functional MMR genes. But somewhere along the way, in a single colon cell, the MLH1 gene got silenced. It wasn't mutated, it was just turned off. This is what we call sporadic MSI.
And that leads us directly and perfectly into our third pathway, because sporadic MSI is almost always caused by CMP.
Exactly. CMP is the bridge that connects these ideas. CMT stands for CPG island methylator phenotype.
Okay, this is the one that always confuses students, myself included. Let's slow way down. What is a CPG island and why are we methylating it?
A CPG island is just a region of DNA that is very rich in the bases cytosine and guanine right next to each other, so CPG.
Okay, just a physical feature of the DNA sequence.
Yes. And these islands are very often found in the promoter regions of genes. The promoter is the landing pad, the on-ramp where the machinery to read a gene assembles.
So it's the start line for a gene.
Perfect. Now, methylation is what we call an epigenetic mark. The cell can physically attach a methyl group, a carbon and 3 hydrogens, onto the cytosine bases in that CPG island.
And what does that do?
It acts like a chemical padlock on the promoter. If you methylate the CPG island, the transcription factors can't land, the machinery can't assemble, the gene is effectively silenced, it's turned off.
So CIMP, the CPG island methylator phenotype, is a condition where the cell just goes crazy with these padlocks.
You've got it. It's a hypermethylation state. For some reason we don't fully understand, the cell's methylation machinery goes into overdrive and starts padlocking promoters all over the genome.
And one of the most important promoters of padlocks is?
The promoter for the MLH1 gene, the main spell checker.
So let's just trace that logic one more time to make it crystal clear. A cell develops the CMP phenotype.
Right.
It then methylates the promoter of the MLH1 gene.
Padlocks it shut.
So MLH1 protein is not made.
The spell checker disappears from the cell.
And without the spell checker, you get thousands of typos in microsatellites. You get microsatellite instability.
You nailed it. That is the exact sequence of events.
Yeah.
And that is why the majority of MSI high tumors we see in the clinic are actually CMA tumors. They are sporadic. Lynch syndrome is the less common inherited cause.
And this brings us to another key concept, the serrated pathway. When we're talking about CMP, we aren't talking about those classic adenoma polyps anymore, are we?
No, we are not. This was a huge shift in the world of pathology over the last 20 years. We used to think all colon cancers came from those smooth, raspberry-like adenomas.
The ones on the AK's-P path.
Right. But pathologists started noticing these other kinds of polyps that looked different under the microscope. They looked like the teeth of a saw blade. They were serrated.
And for a long time, we thought these were harmless, right? We called them hyperplastic polyps.
Exactly. We told gastroenterologists, don't even bother removing them, they're benign. We were profoundly wrong.
What are they then?
We now know that a specific subtype, the sessile serrated adenoma, or SSA, is the direct precursor to this CIMP pathway cancer.
And they're dangerous because?
They're scary because they are flat. They're sessile. They don't grow out on a stalk like an adenoma. They are hard to see on a colonoscopy because they blend into the normal folds of the gut. A gastroenterologist can scan right past one.
And they have a specific driver mutation that's different from the other pathways.
Yes, this is the signature. The classic APC pathway loves KRAS mutations. The CIMP serrated pathway loves BRF mutations.
Specifically, the BRF V600E mutation.
The very one. It's a powerful driver of that same MPK signaling pathway that KRAS uses, just one step down the ladder.
So this gives us a really important clinical tool. Let's say I have a patient with a tumor that tests as MSI high. How do I know if it's Lynch syndrome, which is hereditary and terrifying for the family, or if it's just a sporadic CMP tumor?
You test the tumor for that BRF mutation. It's the tiebreaker.
How so?
If the tumor has a BRF V600E mutation, it is almost certainly a sporadic CMP-driven cancer. For reasons we don't fully understand, Lynch syndrome tumors basically never have BRF mutations.
That is a golden clinical rule, a genuine pearl. So BRF mutation plus MSI high means it's sporadic, and there's no need to panic the family about genetic testing.
Correct. But if the tumor is MSI high and it's BRA wild type,
meaning no BRAF mutation. Then you have to have a very high suspicion for Lynch syndrome, and that patient needs an immediate referral to genetic counseling. That single algorithm saves lives and prevents future cancers in their relatives.
Okay, let's pivot to the so what, the why should I care part of this. We've crashed the car in three completely different ways. We have the broken brake, chromosomal instability pathway,
The APC
the
pathway,
typo-explosion pathway,
MSI,
and the epigenetic silencing, or hacker, pathway.
CIMP.
How does knowing which pathway a patient's tumor took change the way we treat them, especially if they have metastatic disease?
This is where we move from interesting biology to life and death decisions, and the biggest story here is immunotherapy.
Checkpoint inhibitors, drugs like Keytruda, which is pembrolizumab, or Opdivo, nivolumab.
These drugs have absolutely revolutionized oncology for melanoma, for lung cancer, but for years they were a total failure in colon cancer. It just didn't work.
Until we looked closer.
Until we stratified the patients by their molecular pathway. We realized these drugs only work, but work spectacularly well for one specific subtype.
The MSI high group.
Correct. And if you think about the biology we just discussed, it makes perfect sense. In a classic APC pathway tumor, the genome is broken and rearranged, but the proteins it's making are mostly normal human proteins.
So the immune system looks at it and says, that's me, that's self.
It ignores it. The tumor is immunologically cold, but in an MSI tumor.
You have thousands of those frame shift mutations.
And those frame shifts create thousands of completely novel junk proteins. We call them neoantigens. To the immune system, the tumor suddenly looks like a foreign invader. It looks like a virus or a bacterium. It's immunologically hot.
So why doesn't the immune system just kill it on its own then?
It tries. And that's why if you look at these tumors under a microscope, they're packed with lymphocytes, with T cells. The army has arrived. But the tumor has a defense mechanism. It puts up a shield.
A molecular shield.
It expresses a protein on its surface called PD-L1. This protein effectively shakes hands with a receptor on the T cell called PD-1, and that handshake delivers a message, don't kill me, I'm friendly, go to sleep.
And the immunotherapy drug.
The drug pembrolizumab is an antibody that physically gets in the way. It slaps that hand away. It blocks the handshake.
So the T cells wake up.
The T cells wake up, see all those crazy neoantigens again, and launch a furious attack. The response rates can be incredible, with durable remissions in metastatic disease, but only in MSI tumors.
So if you give this drug to an APC pathway patient, a microsatellite stable, or MSS patient,
You are wasting $150,000 giving them potentially serious autoimmune side effects and getting zero clinical benefit.
But testing for MSI or MMR status isn't just academic. It is the absolute first decision point for treatment in advanced disease.
Non-negotiable. It's standard of care.
What about the other big group, the APC KRAS pathway? We have targeted drugs that hit the EGFR receptor, right? Cetuximab, panitumumab.
We do. But this brings us right back to our relay runner analogy for KRAS. Cetuximab works by blocking the EGFR receptor on the cell surface. It stops the growth signal from ever getting into the cell in the first place. It stops the race before it starts.
Which should stop the cancer from growing.
It should, unless you have a KRAS mutation. Remember, the KRAS mutation is downstream of the receptor.
It's the runner who keeps running without ever getting the baton.
He's gone rogue. So if I block the receptor upstairs with cetuximab,
KRAS doesn't care. It's already turned on, and it's going to keep signaling for growth no matter what.
it's completely futile.
Yeah.
So if a patient's tumor has a KRAS mutation, you cannot use EGFR inhibitors. It will not work. In fact, some data even suggests it can make things worse.
So the KRAS wild-type status is the ticket to getting cetuximab.
It is the required biomarker. This is precision medicine in action. We are not guessing anymore. We are looking at the schematic of the crashed car and choosing the mechanic who specializes in that exact type of failure.
I want to touch on one more really important clinical correlation from the outline. The right versus left divide in the colon.
Yes, this is a huge concept.
We mentioned the napkin ring on the left side, which is the APC pathway.
Yeah.
What about the right side?
The right colon, the ascending colon and the cecum, is actually embryologically distinct from the left side. It comes from the midgut, whereas the left colon comes from the hindgut. They are different organs in a way.
And they tend to get different types of cancer.
They do. Right-sided tumors are much more likely to be from the MSI or CIMP-serrated pathways.
And they look different, not just molecularly, but physically.
Completely different. They tend to grow as exophytic or polypoid masses. They look like cauliflower or mushrooms growing out from the wall into the open space.
So they don't tighten and constrict like the napkin ring tumors.
No, because the right colon is wide and capacious and the stool is still liquid over there, so they rarely cause an obstruction. They just grow and their surface is fragile, so they bleed.
A slow, chronic, occult bleed.
Exactly. A little bit every day. Not enough for the patient to see, but enough over months to lead to the classic clinical presentation.
Which is iron deficiency anemia.
Iron deficiency anemia in an older person. This is a clinical maxim you cannot forget. Iron deficiency anemia in an older man or a postmenopausal woman is a gastrointestinal malignancy until proven otherwise.
Even if they have zero bowel symptoms, no pain, no change in habits.
Especially if they have no bowel symptoms, the tumor can get absolutely huge without blocking the pipe. The only sign is the slow blood loss showing up on their lab work as anemia and fatigue.
So let's just recap that clinical landscape.
Please.
Right side of the colon, more likely to present with bleeding and anemia. The underlying biology is often MSI, CIMP, the serrated pathway, and BRAF mutations.
Perfect.
Left side of the colon, more likely to present with obstruction, pain, and altered bowel habits. The underlying biology is often the classic APC pathway, chromosomal instability, and KRAS or TP53 mutations.
That is the general rule of thumb. There are always exceptions, but that pattern holds true the majority of the time.
We've covered a massive amount of ground here. We've gone from once signaling all the way to checkpoint inhibitors. But I want to circle back to something you mentioned right at the start, systems engineering.
Okay.
When we look at CMP, that third epigenetic pathway, doesn't that offer a different kind of hope that the other two pathways don't?
It absolutely does. And this is the frontier. This is where the research is going. Think about it.
Okay.
In the APC pathway, the genes are often physically deleted. A chunk of the chromosome is gone. You can't put it back.
It's a hardware failure. The chip is broken.
Right. In the Lynch MSI pathway, the MMR gene is mutated. The code is permanently broken. But in CMP,
The gene is physically intact. It's still there, perfectly written. It's just silenced by those methylation padlocks.
that's a software lock, not a hardware failure.
And software can be unlocked.
Exactly. And we have drugs, we call them hypomethylating agents, that can strip those methyl groups off the DNA. We already use them successfully in blood cancers like myelodysplastic syndrome, drugs like azacitidine.
So theoretically, you could treat a CMP-driven colon cancer by reawakening the silenced MLH1 gene.
Theoretically, yes. The challenge is delivering these drugs effectively to solid tumors and getting the timing right. But the concept is absolutely dazzling.
It changes the entire goal of therapy.
It does. Imagine turning a malignant metastatic tumor back into a cell that can repair its own DNA, a cell that can undergo apoptosis again just by flipping a few chemical switches. It shifts the paradigm from kill the tumor to reprogram the tumor.
That is a profoundly powerful thought. It implies that cancer isn't always this one-way street of inevitable destruction. Sometimes it's just a locked door.
And we are getting much, much better at picking the locks.
Before we close out, let's do a quick, ultra-simple synthesis. If a listener is asked to explain the difference between these three pathways at a dinner party, assuming a very, very nerdy dinner party, how do you distill it down? Okay, the dinner party summary. I'd say this. Pathway 1 is the traditionalist. It follows the rules of anatomy. It starts as a classic polyp. It breaks the brakes, APC, and it smashes the car's frame physically. Chromosomal instability. It lives on the left side.
The traditionalist. I like it. What's pathway 2?
Pathway 2 is the sloppy typist. It can't spell check, MMR failure, so it creates a million typos, MSI. This makes the tumor look weird and foreign to the immune system, so it gets attacked.
Yeah.
It's the one that responds to immunotherapy.
The sloppy typist. Okay,
Right.
and pathway 3?
Pathway 3 is the hacker. It's sneaky. It uses the backdoor serrated route. It uses a BRAF mutation as its exploit. And instead of breaking the security system, it just uses methylation to silently switch it off. CIMP. It lives on the right side.
Traditionalist, sloppy typist, and hacker. I think that sticks. That's brilliant.
It's the best way I've found to keep them straight in my own head.
This has been an incredibly dense deep dive, but I honestly feel like we actually cracked that cognitive bottleneck. We didn't just list the genes, we watched the whole machine break piece by piece. And that's the goal. Once you see the mechanism, once you understand the story, you don't have to memorize it anymore. It just makes sense.
Well, thank you for guiding us through the machinery.
Anytime. It was a pleasure.
And to our listeners, thank you for diving deep with us. Do check the show notes for diagrams of the WANT pathway and the serrated route. Seeing the visuals really helps lock this in. We'll see you on the next deep dive.